The cell cycle is responsible for cell growth, specialization, and most importantly, replication. In order to understand this, we must look at mitosis and meiosis. Chromosomes are the most important players in mitosis and meiosis. Throughout the cell cycle, a single-stranded chromosome is replicated and becomes a double-stranded chromosome consisting of two sister chromatids. The sister chromatids bind together in their centromeres, creating a chromosome. Before a cell enters mitosis and the M phase of the cell cycle, it spends most of its time in interphase, which includes the G1, S, and G2 stages. Only after the chromosomes are replicated and the cell is made ready for division does it enter the M phase, where mitosis takes place. In the M phase, a cell goes through prophase, metaphase, anaphase, telophase, and finally cytokinesis, when two new cells are formed and the cycle continues. Now let's take a closer look. A cell in interphase begins in G1, where it makes a decision to enter the cycle and prepare for division. The S phase of interphase is where the DNA is replicated in a process called DNA synthesis. Each chromosome now has an identical sister chromatid, and in G2 of interphase, the cell is finally made ready to enter the M phase and begin mitosis. In interphase, it is difficult to see the chromosomes. However, as a cell enters prophase, the first part of mitosis, the chromosome becomes more visible as they continue to condense. During this phase, the nuclear membrane breaks down and the mitotic spindle starts to form between two centrosomes. As the spindle stretches and the centrosomes move towards opposite ends of the cell, the chromosomes move to the center of the cell in the stage called metaphase. In metaphase, the longest stage of mitosis, the chromosomes line up along the middle of the cell, held there by the microtubule spindles attached at the centromeres. The cell then enters anaphase, during which the centromeres of the chromosomes separate and each sister chromatid becomes its own chromosome. The two separated chromosomes are then pulled towards opposite poles as the microtubule spindles shorten. There are now twice as many chromosomes as there were in the previous stages. As the chromosomes gather at opposite poles, telophase begins and two daughter nuclei begin to form, each with identical chromosomes. The nuclear membrane starts to reform and the chromosomes become less condensed. While in the process of telophase, cytokinesis begins to divide the cytoplasm. In cytokinesis, the cytoplasm pinches tightly together in the middle of the cell and eventually separates into two separate cells. Organelles and other materials are then restored in both cells, and mitosis is complete, resulting in the creation and formation of two diploid cells, or cells with two sets of chromosomes. Now let's see where meiosis differs. Meiosis is a process of cell division that reduces the number of sets of chromosomes from 2 to 1. The steps of meiosis are very similar to those of mitosis, however, after the chromosomes are replicated, the cell divides not only once, but twice. Therefore, meiosis is split up into meiosis I and meiosis II. Similar to mitosis, the cell begins in interphase, where the chromosomes are duplicated. In meiosis I, homologous chromosomes separate, whereas in meiosis II, sister chromatids separate. After interphase, prophase I begins, where homologous chromosomes, or chromosomes of the same size and gene, pair up. The homologous chromosomes are bound and held together at a region called a chiasmata. These bound chromosomes form a tetrad. During prophase I, the centrosomes also begin to form spindles. A cell in meiosis usually spends 90% of its time in prophase I. Metaphase I is very similar to metaphase of mitosis, in which the pairs of homologous chromosomes line up along the metaphase plate, held in place by microtubule spindles attached to their kinetochore. In anaphase I, the tetrads separate and the chromosomes move towards opposite poles. Each homologous chromosome is still made up of two sister chromatids. Telophase I and cytokinesis then begin, as each half of the cell has a haploid set of chromosomes. Simultaneously, the cytoplasm pinches together and forms two separate daughter cells in which the chromosomes are still double. After this, the chromosomes are not replicated again and the two new cells begin in prophase II. Here, another spindle apparatus forms and the chromosomes, still composed of two sister chromatids, drift towards the center of the cell. In metaphase II, the chromosomes are again positioned along the center of the cell, attached to spindles. In anaphase II, the centromeres of the chromosomes finally separate, forcing the sister chromatids to come apart and move toward opposite poles. And in telophase II, cytokinesis occurs again as well. Two separate nuclei form in each cell, resulting in 4 haploid daughter cells, each containing a single set of chromosomes, and the process of meiosis is complete. Let's look at the bigger picture and compare mitosis with meiosis. In both mitosis and meiosis, DNA replication occurs during interphase before mitosis and meiosis I actually begin. Mitosis only consists of one cell division, while meiosis consists of 2. Therefore, mitosis results in 2 diploid daughter cells that are genetically identical to the parent cell. Meiosis results in 4 haploid daughter cells, each genetically different from the parent cell. Now, why is cell division important? Well, mitosis enables cell growth and tissue repair as well as replacing dead and damaged cells throughout the body. Meiosis produces gametes and enables genetic variety among them, playing a very important role in sexual reproduction and inheritance.